Hello, everyone. Greetings from Argentina. I wanted to thank Chiara and Ole for inviting me to give my talk here, and also the Martinos Center for giving me the opportunity to share some new data from the lab with you people and hopefully get some exciting feedback. All right, so let's start. In the lab, we're interested in motor learning, and in particular in trying to understand the neural mechanisms supporting memory formation. There are two main types of motor learning that have been studied in the lab thoroughly in the last few years. One of them involves the acquisition of a new motor skill, like we can see here when you're learning to ride a bike, which could be difficult, but also very rewarding. This kind of motor learning involves the acquisition of a new motor program. And in contrast, another type of motor learning that has been studied a lot in the lab is sensory motor adaptation. In this case, in the example we see here, you don't need to learn to ride a bike again to ride a tandem bike, but you just have to recalibrate the existing motor programs that allow you to ride a bike. So in a sense, sensory motor adaptation is a type of learning that allows you to preserve your performance in front of the environmental perturbations. So other examples to give you a better grasp of it is if you now move to a city where the sidewalks are very slippery and you have to learn to adapt to walk on the ice. If you need to play tennis, for example, or serve when the day is very windy. But also sensory motor adaptation allows you to adapt to internal perturbation. And this a good example of this is what happens during ontogeny, as your body changes and the muscles and your body grows, and your brain has to adapt to these changes. You also adapt, for example, to a new prosthetic device, or you can also adapt to brain injury. So the idea that I want to give you here is that these two types of motor learning are very much interdependent, and sensory motor adaptation is crucial to keep precision in motor control. So without sensory motor adaptation, you cannot acquire any new motor skills. All right, so how do we study these two types of learning in the lab? They have two main experimental paradigms that are being used thoroughly in the lab. One of them is motor sequence learning. So in motor sequence learning, you learn to perform a sequence, like in this case, with your fingers. You could have the sequence like it is here on the screen, and you just have to tap the sequence as accurately and as fast as possible. And then you construct these sort of learning curves where you see online gains, but you could also see offline gains overnight. In the case of adaptation, there are many different experimental paradigms. And they also depend whether you are adapting to a visual perturbation or a proprioceptive perturbation. This is one setup that we use very much in the lab. And in this case, what subjects have to do, what they can see here is a start point and then a cursor, and a target. And they manage a cursor using a joystick. So it's just to give you a better idea what's happening during this paradigm. This is the target the subject is seeing, and this is the start point. And initially, in a baseline condition, what subjects do is they're instructed to, throughout the whole task, to hit the target with the cursor they control with the joystick. So in initial condition, there is no perturbation, and you can see that both the cursor and the hand move in the same direction. And this would be if you plot the trajectories for, let's say, pointing movements to 8 targets instead of 1. This is what your trajectories would look like. Now then you introduce a perturbation. For example, in this case, you can see this is just an optical rotation, like 45 degrees to the left. And so the hand keeps going initially to the target, but the cursor is going deviated 45 degrees. And this would be what the trajectories would look like. But later on, if you keep practicing, this may take about 30 or 35 minutes. What happens is you learn, and so you learn to bring the cursor to the target. And as you can see, the trajectories look pretty much like they look in the baseline condition. So Whereas in the case of motor sequence learning, you see a benefit in your performance. Here, what you see is the recovery of the original performance after the perturbation. All right, and in motor adaptation, you don't see offline gains like you do see in motor skill learning. So please interrupt me if there's anything that isn't clear enough, okay, before I go on. All right, so what we're interested in the lab is in two things. On the one hand, we're interested in trying to study the differences between these two types of motor learning, but also the commonalities. And we think this is important because of what I told you before, because the two types of learning are very interdependent and you need one for the other. And the other main aim that we have in the lab is trying to understand the mechanisms of the basis of memory formation, but trying to see whether these mechanisms are share some features with what is known from the declarative field. And this is because there have been a lot of several studies in the last few years that are sort of suggesting that there isn't such a clear cut or separation between the consolidation of motor or procedural and declarative memory. So some of the data I'm going to show you today, you will see that there's a hint that there are some of the mechanisms involved in memory formation are common to both procedural and declarative memories. So the way I organized my talk today is I'm going to focus on, it's divided in 3 parts, and each of these 3 parts relate to one main line of research in the lab at the moment. Okay, so one of the interests we have is trying to understand motor memory consolidation during the day. And I'm just going to show you a couple of experiments that are aiming at trying to unveil the time course of consolidation in sensory motor adaptation. Another issue that the lab is interested in is trying to see whether these memories that consolidate during the day suffer any additional modulation during sleep. And finally, we're also interested in trying to understand how these changes that occur with memory formation impact at the level of structure in the brain. All right, so let's start with the first point. So the time course of memory consolidation has been studied for many years in the declarative field, and in the motor field, I would say like about less than 10 years ago, we know that for motor skill learning, it takes about 4.5 to 5 hours for a sequence to consolidate. But in the field of sensory motor adaptation, it hasn't been very clear. In general, when you're trying to use or to unveil the time course of consolidation behaviorally, people have used retrograde interference protocols. But the retrograde interference protocol has failed in the field of when you're trying to understand, to unveil the time course of memory consolidation in sensory motor adaptation. We're not exactly sure why it is, but this was an interest of the lab. So what we thought is, well, if retrograde protocols fail, why don't we try anterograde interference protocols? And this is the first study that we run. This is a collaboration we started many years ago now with the lab of Reza Shatner at Johns Hopkins University. And this study was carried out by my student Gonzalo Lerner and Reza's student Scott Albert. So what we did was the following. Subjects have to adapt to 2 visual motor perturbations. Like I said before, these perturbations involve an optical rotation. But in here, what they did was they first adapted to perturbation that was rotated, let's say 30 degrees in one direction. And then after a certain time interval, they adapted to the opposite perturbation. Then we waited 24 hours and we measured the memory retention. Now, we have 4 different experimental groups, and the groups differ based on how long was the interval between learning perturbation A and perturbation B. And this was 5 minutes, 1 hour, 6 hours, or 24 hours in between the 2 perturbations, okay? And like I said, after the second adaptation to the second perturbation, we waited 24 hours and we measured the retention. And then as a control group, we had a group that was not exposed to the first perturbation, but was only exposed to B. Okay, so basically, what I'm showing you here is the same experimental design. And what you can see down here is the learning curves. We plot the pointing angle, which is, you can see they find it reaches an asymptote towards a halfway practice. And each color represents a different interval group. And the one in gray represents the control group. So what you can see is that all the 5 groups are very similar when they adapt to the first perturbation. Then there's these different intervals. And what you see is how the performance is affected when they adapt to the second perturbation. And so we computed 2 different measures here to get an idea of how the memory of second perturbation was being affected by the first one, by the consolidation of the first one. And one of the things we did was fit these curves with a single exponential function, and we simply computed the beta weights, which give us an idea of the speed of learning at which they are learning the second perturbation. And this is what I'm showing you up here. So if you plot this beta weight as a function of the different groups and compare it with the control group, what you could see is that both the groups that were separated by 5 minutes and 1 hour were affected in their ability to learn the second perturbation. And by around 6 hours, there was no significant difference from the control condition. Okay, this relates to the ability to learn. What happens to the long-term memory or memory retention? So when we look at that, this is what we found. So if you look at the percent of retention, and this is percent because it's relative to the asymptotic value, what you see is also both the 5-minute interval group and the 1 hour interval group were very much affected by having learned A initially. But about 6 hours for the 6 hour interval group, there was already no significant difference from the control condition. So what we can conclude from this experiment is that both in relation to the ability to learn and the memory retention, which would be long-term memory because we measured this 24 hours later, they're both affected and they both start to get released from interference at about 6 hours. So we're guessing that the time course of memory consolidation for visual motor adaptation is about 6 hours, which is consistent with what sort of consistent or close to what has been shown for motor skill learning. Okay, so we got an idea of this time window, and then we asked, what is the network that may be related to this process of consolidation? And so what we did, this is work that was carried out in my lab by Jorge Vichalta and myself. And what we did is we used a very simple resting state paradigm, and we had people being scanned before and at several time points after training on visual motor adaptation task. And a different group, that was a control group that did exactly the same thing, but was not imposed a perturbation, okay? And we obtained 2 measures of memory retention. A short-term memory retention measure that was obtained about a minute after they finished training. This training takes about, to give you an idea, 30-35 minutes. And then the day after, 24 hours later, we obtain a long-term memory retention, okay? And at all these time points that I'm showing you here, we obtain resting state data, and we computed the functional connectivity to see if we could capture any changes that would relate to the long-term memory formation. And so what we found is, you can see here for the visual motor adaptation group and the control group, these were the areas that showed an increase as a function of time. And we're talking here about M1 premotor cortices, posterior parietal cortices, and cerebellum. And most of them were contralateral to the trained hand, that was the right hand, except for the cerebellum that we saw increasing connectivity in bilaterally. As you can see in the control group, there was a slight increase for the 3 hours group, but then this decay. And something that is not in this analysis because it, well, I didn't include it because it wasn't significant, but it's quite interesting, and that's why I will mention it. This strong connectivity that we see at 5.5 hour decays at 24 hours. So that was interesting, and this is something that we have replicated many years ago in another functional study that we carried out when I was in Toronto. Okay, so the next question is, okay, so if this increment in the functional connectivities is at all related to consolidation, then we would expect it to predict at the level of retention the day after. And so what we did was correlating the strength of the functional connectivity of this pattern that I'm showing you here as a function of short-term retention and long-term retention. And if you focus on the long-term retention first, you would see that those subjects that retain the most are the ones that show the strongest connectivity of this network. But interestingly, the opposite pattern is was found for the short-term retention. That, as you remember, we obtained this measure about a minute after subjects finished training. So in these cases, those subjects that show stronger connectivity of this pattern here show less short-term memory. So this makes us think that there may be like two different networks involved in the formation of short-term and long-term traces, right? And this may somehow relate, I suppose, to the existence of a more explicit network that is not being captured here by this multivariate analysis that we carried out. Okay. So to summarize this first part of my talk, we found that visual motor adaptation memories appear to consolidate within a time window of about 6 hours. This timing is in line with that described for motor sequence learning. Memory consolidation is associated with an increment in the functional connectivity of a network, including the bilateral cerebellum, left parietal, and left sensorimotor cortex. And the fact that this network correlated positively with long-term memory, but negatively with short-term memory, points to the existence of two independent processes supporting memory formation. Good. Okay, so the second issue that we are interested in the lab is, do these memories that consolidate suffer any more modulation during sleep? And if so, what are the mechanisms involved in this consolidation? So what do we know about sleep-dependent consolidation of motor memories? We know that sleep benefits motor sequence learning, and we also know there's evidence that it benefits visual motor adaptation, although there is some controversy about this. We also know, in terms of the mechanisms involved in sleep-dependent consolidation, that in declarative memories, the precise coupling between the slow oscillations and the spindles is critical for sleep-dependent consolidation. So we wonder whether this coupling may also be important for the consolidation of motor memories. And so what we did is we conducted an experiment. This was carried out by Agustin Solano in my lab and with Kelme also in my lab, in which we had a set of subjects that were tested, that were trained on a visual motor adaptation test, the same paradigm I showed you before, or a control condition that differ only in the fact that the perturbation was not applied. And these two training sessions took place 1 week apart and in a counterbalance order. And so what we did, subjects train right before going to bed, and then they sleep in the lab. They slept in the lab. We monitor the EEG activity in these electrodes that I'm showing you here. And then the next morning, they woke up, they had breakfast, and we tested them to get quantification of memory retention. Okay, so the first thing that we looked at was whether the motor learning influenced the density of sleep spindles. And what you can see here is a percent change in the density of spindles. By percent change, I mean how much the density of spindles increase when you learn compared to the control condition, okay? So 0 would be that they are similar. And what we find when we are comparing non-REM 2 with non-REM 3 is this increment in the density of spindles in non-REM 3, and particularly for the left hemisphere when you compare it to the right hemisphere. And when I talk left and right hemisphere here, I'm comparing the electrodes in violet with the electrodes in green. We're not including the midline, okay? So if you actually look at the spatial distribution of this, what you see in these topographic plots is this would be, now this includes all the electrodes, this would be the distribution of the density for the visual motor adaptation condition, the control condition, and this would be the percent change in visual motor adaptation compared to the control. So what you can see, this corresponds to this graph here. What you can see is that there's a nice increase in the density of spindles in the left hemisphere, which was the hemisphere contralateral to the training hand. That was the right hand. Good. So the next question is, what kind of spindles are the ones that being influenced by learning? Because we know from the declarative literature that the fast spindles are the ones that seem to be more involved in memory consolidation. So we basically look at that aspect, and we actually discriminated between fast spindles that are now plotted here in gray and slow spindles that are plotted in black. So I'm showing you here again the spindle density percent change of VMA relative to the control condition. And what we see again is a modulation at the level of non-REM 3, and a very strong modulation on the density of the fast spindles compared to slow spindles. And if I show you the spatial distribution of this, what you can see is the fast spindles on the top panel. You can see this nice origin from around like parietal areas, and for the slow spindles and more frontal areas. But when you actually look at the percent change, which would mean that the learning related change in the density of fast spindles, what you see is that there is an increase for the fast spindles that is not there for the slow spindles. And what you see here is the difference for when you compare the left versus the right hemisphere. As I mentioned here, comparing the violet versus the green. Although here you can also see some increase in frontal areas. Okay, but so far what we can tell is that visual motor adaptation is modulating the density of spindles, and in particular of fast spindles during non-REM 3, and in a way that seems to be lateralized, okay, or contralateral to the train hand. All right, so the next question was, what happens with the coupling between these fast spindles and the slow oscillations, which is what we have found, what people have found in the declarative memories, in the consolidation of declarative memories, to have a preponderant role. So what I'm showing you here is the following. In the top panel, what you can see is the density of slow oscillations, which is what we first looked at. For the learning condition, the control condition, and the difference, the percent change between the two, as you can see, there was no modulation of the density of slow oscillations during non-REM 3. But when you look at the slow oscillations coupled with fast spindles, what you can see is that there is quite a strong modulation at about like 30% change when you compare it to the control, and that is quite nicely contralateral to the trained hand, okay? And this is just to show you that this only applies for the spindles, for the fast spindles that are coupled with slow oscillations, and not for the slow spindles coupled with slow oscillations, which show you like this distribution and no significant effect. Good. And so what we get from here is that visual motor adaptation learning seems to modulate the coupling, not only the density of fast spindles, but also the coupling between slow oscillations and fast spindles. But the question here to try to understand whether this relates or not to memory consolidation is whether this can predict the level of memory retention the day after. And when we look at that, what we found is that there was a significant correlation between the density of slow oscillations coupled with fast spindles and the level of retention. But this relationship was not there when we correlated the uncoupled spindles with the level of retention. So this is nice because it's basically telling you that only coupled spindles are the ones that can predict long-term memory and not uncoupled spindles. Good. So to summarize the second part of the talk, we can conclude that visual motor adaptation increases the density of fast spindles, specifically during non-REM 3. Visual motor adaptation increases also the coupling between fast spindles and slow oscillations locally over the contralateral hemisphere to the trained hand. And only those spindles, the fast spindles that were coupled to slow oscillation and not the uncoupled spindles predicted long-term memory.
Valeria, we have, sorry to interrupt you. We have a question now, a quick question. Bruce, you can ask the question yourself if you want.
Oh, okay, just a very obvious question. Do you get kind of the same pattern of consolidations and timings if you use your non-dominant hand for the motor training?
I don't know. We haven't looked at that, but I would expect so. I don't know. Okay.
I think so, yeah.
All right. So the third issue that we're interested in the lab, as I said, was structural plasticity. And here I would like to switch back now to motor sequence learning. I mean, so far I've been showing you data that was more related to sensory motor adaptation, partly because it has been, those aspects I've told you have been more investigated in motor skill learning. But a few years ago, we started a collaboration with Julien Vaillant and other people, and we started looking at some aspects of motor sequence learning that we got interested in. So as I showed you before, in motor sequence learning, you have like online games that take place while you're practicing the sequence during the training session. But then the day after, you also see offline games. And in general, these offline games are thought to depend on sleep, and they're thought to depend on the processing of the hippocampus. Now, more recently, Ingrid Bonstrup from Leo Cohen's lab described another type of offline games that are very intriguing, and they are called micro offline games. And they take place if this is the learning curve that you get from motor sequence learning. They take place especially in the first part of learning before reaching the asymptote. So if we zoom in here, what we can see here is the tapping speed as a function of trials in this portion only. And so what Ingrid realized was that when you look at the gains that you get during this initial training phase, there seems to be more gains in the resting periods while the subject is not practicing than while the subject is practicing. So what she did is she computed two different metrics, what she called the micro online gain, which is basically the performance changes or the gains in performance from the first if you look up here, the first tapping to the last tapping. And she also computed the micro offline gain, which would be the gain in performance from the last tapping to the next tapping after the break, okay? So in this case, in this paper, I think she did, the protocol was 10 seconds of tapping, 10 seconds of rest, 10 seconds of tapping, 10 seconds of rest. Okay, and so if she now plots, again, always keep in mind that it's during this initial period. This micro offline gains, the total amount of learning, which are in black, and the micro online gains, what you can see very clearly is that the total amount of learning is pretty much explained for by the micro offline gains. This is another way of portraying the same thing. And what this tells you is that this is the total amount of learning. This is the gain that you get from micro offline gains. And this is what you get from micro online gains, which is pretty much 0. So this is interesting. As I said, it differs from the other offline gains I told you before that occur overnight, because this takes place in a much faster timing. What they suggested was that this may be a sort of like a fast consolidation. But I think the also interesting or possible interpretation of this is this may be some sort of like reactivation that takes place during these quiet rest periods in a way that is reminiscent of what happens in rats while they're learning a maze, right? And so, as you know, in the play cells of the hippocampus, you see this neural replay during the rest while the rat is learning a maze. And so we thought, well, maybe this is what's happening here. Maybe it's possible that the hippocampus not only takes place in processing offline gains, but it may also be important for this initial learning. And maybe there is some reactivation going on in these rests in which the hippocampus is also involved. And so this is what we investigated in this study. So in this study, we obtained several, we scanned people with several different sequences, but the results I'm going to show you today only involve the fMRI and the diffusion data. And so we were interested in trying to understand whether the hippocampus is involved in the production of these micro offline gains that I just showed you. So what we did is we used fMRI to examine whether the hippocampus increased activity during these quiet rest periods, interleaved with the practice. And then we used diffusion weighted imaging, and in particular, we apply the DTI model, and we focus on a particular image or metric, which is the mean diffusivity. Why we use mean diffusivity? Because Janiv Asaf's lab have shown years ago that mean diffusivity is an interesting proxy for plasticity. And in particular, what he showed is that when you have, when you learn, and this study was carried out in rats and in humans, and what they saw is that in rats, the learning is associated with a decrease in the level of mean diffusivity, and this decrease in the level of mean diffusivity is associated with an increase in glial hypertrophy, particularly astrocytic hypertrophy. So the idea being that these structural changes that occur with learning, because you can block these changes if you use an NMDA antagonist. So this decrease in mean diffusivity reflects this LTP-like plasticity that is revealed partly as an increase in astrocytic hypertrophy. Okay, so this is our proxy of structural plasticity, which is not the best model that one can find, because there are, I mean, nowadays there are better models that are coming out, but unfortunately, those models like the one by Palombo and others are, but even though there are better models, that they can, today you can only apply it to data acquired at very high, with very high or potent gradients that most scanners don't have. So for now, what we used was mean diffusivity as a structural proxy. Okay, so this is the study. The study was part of a collaboration with between the Latin American Brain Mapping Network and the Quebec Brain Imaging Network. And so we, Julian Bayon, Jorge Jovicic, and Jorge Armani were involved in the study. And the whole work was carried out by Florencia Jacobacci, a grad student in my lab, and Gonzalo Lerner. And so what we did was we had 20 subjects coming to the lab in different weeks. And they were first obtain a diffusion scan. Then they practiced, they learned the motor sequence task. And we scanned them we obtained like a ball images while they performed the task. Then 30 minutes later, we obtained another diffusion scan, and 24 hours later, another diffusion scan. And then there was a control condition. The same subjects performed the control condition also. The control condition was exactly the same, but the difference was that we used a task that didn't involve, it involved tapping, but didn't involve learning. All right, so this is our behavior. This is just the opposite measure of what I showed you before for von Stroop's study. Here we show the inter-tap interval and how it dropped as a function of practice. We computed the same metrics that von Stroop computed, that is the micro offline gains, micro online gain, micro offline gain, and total learning. And we managed to replicate the same results. So basically, this is the micro offline gains, and micro offline gains explain most of learning. And micro online gain didn't explain learning. And this is another way of portraying the same thing, but the interesting thing in this plot, what you can see is the cumulative gains that occur offline or online, and this is total learning. And what you can see here, this is as a function of blocks, it's the same blocks that you see up here. And the interesting thing is that about half of training is when you get the maximum amount of gains, right? This would be the amount of gains, again, during these quiet rest periods. Good. Okay, so we first look at the results of the fMRI, and we use a typical block design. And what I'm going to show you first is what were the brain areas that were most active during the task and during the rest. And this is not surprisingly, I forgot to say that for this motor sequence learning task, subjects had performed the task with the non-dominant hand, okay? So not surprisingly, we found an increase in contralateral sensory-motor areas, supplementary motor area, cerebellum, and putamen. But the interesting thing is when we computed the opposite contrast and we look at the areas that were more active during the rest than during the task, we found that there was an area in the precuneus and also the left hippocampus. This was, just keep in mind that this was like a whole brain analysis, okay? This was not a region of interest analysis, and it was corrected for multiple comparisons. This was work carried out in collaboration with Abraham Jeffal. Okay, so we thought, well, this is pretty cool. Let's see what we find when we actually look at the first half of learning, in which I show you is when micro offline gains take place versus the second half of learning. And when we do so, in the hippocampus and the precuneus, we see that during the first half or early part of learning, we saw more activity of the hippocampus than during late, and the same thing for the precuneus. And if you want to know whether this activity relates to micro offline gains, then you have to actually correlate the level of volt to the micro offline gains. And this is what we did for the early and the late portions of learning. And what we found is that both for the hippocampus and the precuneus, we saw a significant correlation between the activity in these areas and the level of micro offline gains. So those subjects that show more gains early in learning show more activity in these two areas. But interestingly, this relationship fell for the late learning. So activity in the hippocampus and the precuneus didn't predict a gains in the second part, which would be logical because you find the micro offline gains mostly in the first half. Okay. So then we asked, what happens to the structural plasticity? Is this a very short-term and short-lasting, like working memory-like phenomenon, or short-term memory-like phenomenon that doesn't impact on brain plasticity, or it does? And so, as I said, we use this measure of mean diffusivity to look at that. What I'm showing you here is the result of also a whole brain analysis that we carried out. And what we found is these two regions, also in the precuneus and the left hippocampus, show decreases in mean diffusivity at 30 minutes. What I'm plotting here is basically the percent change in mean diffusivity between the baseline and the 30-minute time point, and between the baseline and the 24-hour time point. And the bars that you can see here, well, these are the means, the bars, the gray bars, but these bars are not error bars, they are the confidence intervals. And this is just to get an idea of whether it's significant or not. If it includes the zero, it's not. So what we can see here for the precuneus is that there was a decrease in mean diffusivity for the motor sequence learning condition, but not for the control. And this persisted for 24 hours. And in the case of the left hippocampus, there was also a decrease in mean diffusivity for the left hippocampus when they performed the task. And this reverted 24 hours later. And when we actually put together the results from the functional analysis in green and the results from the microstructural plasticity analysis of mean diffusivity, you can see that is quite a nice overlap, both for the precuneus and the left hippocampus, although the functional data was also extended more anteriorly too. And when we actually look at the correlation between the two, we found a relationship that is marginal, in which we see that those subjects that show more, that were more, this was carried out for the hippocampus. Those subjects that had the hippocampus more active during training, during the rest periods, also show a stronger decrease in mean diffusivity. Excuse me. Okay, so this would suggest that the same regions that show these increments in activity during the micro offline gains show also changes in microstructure. So to summarize the third part of this talk, we conclude that functional data suggested that the hippocampus and the precuneus were associated with the production of micro offline gains in motor sequence learning. The overlap between the functional and structural results suggests that the same network that reactivates during the quiet rest periods of training also undergoes structural plasticity. And we hypothesize that the hippocampus may be involved in this reactivation or coding of both declarative and procedural memories. And with that, I am going to finish by thanking all my lab. These are a lot of people that I showed you before that carried out this work, and the other collaborators that also contributed to this work. Thanks, everyone.